Okay, so welcome to this next video on bacterial drug transporters. So far we have seen two major families of drug transporters that bacteria use, namely the ABC transporters standing for the ATP binding cassette transporters and the SMR transporters standing for small multi-drug resistance transporters. Now, we're now going to look at some other families. So, let me introduce you to the next one. So let's say this is the phospholipid bilayer. Now the next family is known as the mate family. Okay, so I'll draw it here, whoops. Mate stands for multi-drug and toxic compounds extrusion family. Okay, so these are the multi-drug, or mate for multi-drug and toxic compounds extrusion transporters. So this stands for multi-drug and toxic compounds extrusion transporters. Okay, so that's what a mate transporter is. It stands for multi-drug and toxic compound extrusion transporter. I think I'll get rid of that S there. Multi-drug and toxic compound extrusion transporter. That makes more sense. Right, okay, so again, these work by secondary active transport. So they're going to extrude multiple drugs, so they're going to extrude our antibiotics that we're putting into the cell, or we're trying to get into the cell anyway, and they're also going to extrude toxic compounds. So multi-drug and toxic compound extrusion transporters, again, they are a massive great family of proteins that are in the cell membrane, and they work by secondary active transport, again with this proton gradient. So they allow protons to move back into the cell in exchange for the drug being moved out of the cell, or the toxic compound indeed. Okay, so there's another family which we'll be hearing a lot about. Okay, so let's move on to the next family then now. So the next family, which we'll draw down here, is the MFS family, which stands for Major Facilitator Superfamily. So this is the MFS transporters, and this stands for Major Facilitator Superfamily. So Major Facilitator Superfamily. Okay, and again, these are proteins which you find in the cell membrane of bacterial cells, which are going to extrude drug molecules from the cell, and which will couple the movement of the drug out to the movement of protons in, and that's how they drive the movement out. So again, they are an example of a secondary active transporter. And again, I'll repeat myself because it's worth repeating that the ABC transporters are the only known bacterial drug transporters which work via active transport, or via primary active transport, because both secondary and primary are both active transport mechanisms. So protons come in, drug goes out. So that's the Major Facilitator Superfamily. And now you'll be pleased to know just one more family to talk about, which is the Rnd family, which again we'll be hearing a lot about. We'll look at the structure of these in a lot of detail. So this is the Rnd family, which stands for Resistance Nodulation Cell Division family. So resistance here, resistance cell division, resistance nodulation, Rnd, nodulation cell division family. Okay, so resistance nodulation cell division transporter or family, whatever you want to call them. The Rnd transporters are the resistance nodulation cell division transporters. And again, they are proteins within the membrane of the cell. So here is our Rnd transporter in turquoise. And it will couple the movement of the drug out of the cytoplasm of the cell to the movement of protons in from the extracellular space down their electrochemical gradient that has either been set up by the electron transport chain proteins or by the proton ATPase if that bacterium happens to get its energy from fermentation sources. Okay, so these are the five major families of drug transporters that are made use of by bacterial cells. The ABC transporters, or ATP binding cassette transporters, the SMR, or small multi-drug resistance transporters, the MATE transporters, multi-drug and toxic compound extrusion transporter, and the MFS, major facilitator superfamily, and the Rnd, resistance modulation cell division transporters. Okay, so we will see examples of all of these. To have you prepared for some of the ones we're going to see, Rnd transporters. A nice example of an Rnd transporter is the ACRB transporter. We'll be hearing a lot about that one later on. And the SMR, a nice example to have now ready for an SMR transporter is EMRE. So EMRE is an example of a small multi-drug resistance transporter. Now, just before we end this video, let's have a little look at why these drug transporters are so interesting. Well, let's have a look at E. coli. E. coli has 37 known drug transporters in its cell membrane. So 37 drug transporters. That's a lot, okay? Now, when you look at these drug transporters, they are all loads of them are transporting the same drug. So there is a huge amount of degeneracy in this system. Now, what that suggests is that these transporters are not merely there to transport the drug out. Instead, it suggests that they have other physiological roles and that transporting the drug is just something they happen to do. Okay, which helps for the bacteria to survive, and therefore we see it in the population today. But it suggests that they have other physiological roles. The level of degeneracy that you see in these bacterial drug transporters suggests that. Okay, I also want to draw your attention to the fact that some of these drug transporters are incredibly specific. So let me give you two examples of Rnd, so resistance modulation cell division transporters that are within E. coli's membrane. So two examples of these, and let me draw them here, are the VBDE transporter, so VBDE, which specifically transports phosphomycin out of the cell. So it transports phosphomycin, and pretty much no other drug apart from phosphomycin is acted upon by this VBDE transporter. Okay, so phosphomycin is specifically targeted by this VBDE transporter. Remember, phosphomycin is this antibiotic that inhibits the pyruvyl transferase enzyme in the cell wall biosynthesis pathway. So it prevents the synthesis of N-acetyl muramic acid, basically, from N-acetyl glucosamine. Okay, now another example of an Rnd, a resistance modulation and cell division transporter, is a transporter known as ACRAB. Now this one, on the other hand, contrast this rather to VBDE. ACRAB transports pretty much every drug we know of. So basically, you have drug transporters which are extremely specific, such as VBDE, which merely transports this phosphomycin drug out. And then you have other drug transporters in the same family. These are both Rnd, resistance modulation cell division transporters, that transport nearly all drugs known, basically. Okay, so that's an interesting question. Well, how does it transport nearly all drugs that we know? When we think about, enzymes and proteins, usually what we think of is this protein having a specific little binding site that some specific molecule is going to be able to bind to, that will cause conformational change, and then the drug will be extruded onto the other side. Okay? This is not working like that at all. This is working for nearly all drugs we throw at this. So how does this work? How does it have no specificity? How can any old drug be extruded by this transporter? It's an interesting question, and we want to know the answer. And we will search for it in upcoming videos. So remember, these are both Rnd transporters, so they are therefore moving the drug coupled to the movement of protons moving back into the cytoplasm.